Hi, everybody. Those 5 minutes went a little bit fast, I guess. Um, Adrián, it's now your turn
Hello.
to show us your latest updates. And safe to say, your research that you're going to present it's under review currently, right? So maybe
Yeah,
people will be able
almost.
to read the thing out soon. Looking forward to see how your project is going and
Oops,
the latest
correct.
results. Thank
Yeah. Can you see the screen?
Correct, yes.
Okay. Sorry. Can you see the pointer now?
Pointer, no.
Okay, but now yes.
Yes, no, yes.
Okay, then forgive me if I don't look directly at the camera. But
No
yeah,
problem.
first, congratulations, Valeria, on your very nice presentation and your nice work. So I'm Adrian. I'm a PhD student in Maria Selmer's group at Uppsala University. And now we're going to change the topic and I'm going to present about a molecular mechanism for fusidic acid resistance. First, I will introduce you to Staphylococcus aureus, which is a gram-positive bacteria that can cause infections in humans. It's quite common in about 1/3 of the population. It's present in the skin or the nose. But the thing is, it can be an opportunistic pathogen, for example, if you get a wound or if you are immunosuppressed. And in fact, antibiotic-resistant Staph aureus is one of the deadliest resistant bacteria globally, according to the latest study on the global burden of antibiotic resistance. And thus, effective antibiotics are essential for treatment. One antibiotic that can be used against Staph aureus is fusidic acid. This is the chemical structure of fusidic acid, and this is how you may find it in a cream, for example, in the pharmacy. And it's not effective only against Staph aureus, it's also effective against gram-positives in general, and it has been in used in Europe since the 1960s. Fusidic acid has a unique mode of action because it targets two steps of translation. And when I'm talking about translation here, I'm not talking about translating languages, but it's kind of similar in a way. So what is this translation? If we look at the central dogma of biology, we have our genes stored into DNA, and they have to be to get into proteins to give an effect. And first, the DNA is transcribed into RNA, which is another medium, and this can be read and translated into actual proteins. And this is what fusidic acid targets, the translation step, or the protein synthesis, basically. And translation is performed by the ribosome, which is a nanomachine, a very complicated mix of dozens of proteins and RNA together. And this is a schematic of the ribosome that I will explain briefly so you can understand the rest of the presentation. The ribosome has two subunits, the large subunit here in blue and the small subunit here in yellow. Here we can see the messenger RNA, which is the RNA to be translated into protein. And here in brown are what are called the tRNAs, which are adaptor RNAs that will help bring the amino acids to form the peptide. And the ribosome has three sites where the tRNAs combined, E, P, and A. And then the letters come from A for amino acid, which is where the amino acid comes to join the peptide. Then P for peptidyl, which is where the tRNA carries the peptide, or that will become the protein. And E for exit, where the tRNA shortly stays before leaving the ribosome. And here I have a cartoon representation that us structural biologists like to use to represent atomic structures in a more simplified way. And I will use it probably in the rest of the presentation. And translation is a great target for antibiotics, mainly because it's quite conserved how it works, but it's sufficiently different between, for example, us humans and bacteria that we can target their translation system and not affect ours. And this is just an schematic of all the steps that happen in translation in a simplified way. And you can appreciate all of these are different antibiotics that target different steps. Basically, almost every single step can be targeted by an antibiotic. And in fact, more than half of the antibiotics available in clinic target translation. But for fusidic acid, as I said, targets two steps of translation, namely the elongation cycle here in yellow which is where in each cycle, one amino acid is added to the peptide, and it would target here in a step called translocation, and I will explain briefly later. But it also targets what is called recycling, and this is where the ribosome finishes producing a protein, and it needs to be recycled so it can be reused to produce another protein. And that step is also targeted through fusidic acid. But since elongation cycle happens many times per protein and recycling only one, it mainly affects or inhibits the bacterial growth by blocking this elongation cycle. And as I said, in particular, it blocks the translocation step. And this, I'm showing you again a schematic of the ribosome. Just to remind you, here we have the E site, which is free here, P site, and A site. So a new amino acid comes in with a tRNA on the A site that is brought by a protein called EFTU in bacteria, and then is joined to the peptide here. The thing is, obviously, these tRNAs have to move by one codon. So you have, again, the A site free, and a new tRNA can be joined to give the amino acid. And this job of moving the tRNAs by one codon is done by elongation factor G, which is this protein in green that binds to the ribosome and pushes the tRNA and mRNA by one codon. So then the A site is free again, and a new tRNA can be delivered. And I will call elongation factor G EFG many times. So fusidic acid blocks this step by binding to EFG just after translocation before it leaves here at this step. And when it binds to it, it locks it to the ribosome and it cannot leave. Since EFG is locked here on the A site, new tRNAs cannot be delivered, and the translation is basically stalled. Now, if we look into the different resistance mechanisms of bacteria, just so you understand how this is relevant for fusidic acid, an antibiotic to inhibit the bacteria has to, here in this schematic is shown in green, it has to get into the cell, find its target, which is shown in the schematic in pink, bind to it and inhibit it. So commonly bacteria can maybe not take up the antibiotic and then they will be resistant, or they can pump it out of the cell so it cannot find the target, or they can modify the target or mutate it so the antibiotic cannot bind to it, or for example, modify the antibiotic instead or degrade it so it cannot act. There are other more uncommon mechanisms or more unconventional, let's say. And for fusidic acid, the most common type of resistance is through what is called factor-associated protection or target protection. And here, in this case, the bacteria produce a resistance protein, which is depicted here in gray, that binds directly to the target of the antibiotic and somehow provides resistance. And we'll see how it is for fusidic acid. So in the case of fusidic acid, this type of resistance is called FusP type. And as I said, it's target protection or factor-associated protection. And the resistance protein that does this is called FusP. And this is the crystal structure shown in a cartoon representation. And we say that it has two domains here, an alpha helical bundle, and this C-terminal domain that binds to zinc. So what do we know about FusP? FusP is a small protein, and when it's expressed in these cells, they become resistant to fusidic acid with a minimum inhibitory concentration of 60 micrograms per milliliter, compared to 0.128 for the wild type. And we call this low-level resistance because when we compare it with the other more common type of resistance, which is called FusA, these provide a lower level of resistance. But as I said, FusP is much more common. And FusP, to perform this target protection, it needs to bind to the target, which in this case is elongation factor G or EFG, and it does so with very high affinity, with 60 nanomolar constant of dissociation. And there is an NMR group that has shown that when FusP binds to EFG, it kind of increases its dynamics, and they propose that through this, it makes it more loose on the ribosome, something like that, and then it lets it get released from the ribosome and bypass the inhibition that way. But all the molecular mechanism details were quite unclear. So what we set out to do is to try to figure out how does FUSB rescue EFG. And since we are a structural biology group, the way we try to answer questions is by looking at how the atomic structure looks. And in this case, we wanted to capture FUSB as it binds to EFG on the ribosome before they leave, so we can see exactly what's happening. And this is not so trivial, because if we look again at this schematic of translocation, in this step is where it's locked, right? EFG bound to a ribosome, and here would FUSB come in and release it from the ribosome. But we knew that this happens quite quickly. Obviously, it needs to free EFG out from the ribosome, so the ribosome can continue translating, and this has to be done fairly quickly. And I will come back to this later. But the method that we decided to use for this purpose is cryo-electron microscopy. So here we would use an electron microscope that is basically like a microscope, but instead of using light, it uses electrons to take images. And this is an example of how an electron micrograph may look like. And in fact, this is ribosomes that are locked by fusidic acid and are being rescued by FUSP. And you can see the scale that this is really tiny, just 25 nanometers is more or less one ribosome. And all of these particles that we call are in this case ribosomes. And what we do in cryo-EM is that these are 2D images, but what we want is a 3D reconstruction. Since we have lots and lots of ribosomes in different orientations, if we can combine their 2D images, we can get a 3D reconstruction. So we can pick thousands of them in the images, hundreds of thousands, even millions of them, and then get a 3D reconstruction from this. For example, I show here with EFG. But since we are doing this at a single particle level, at a single ribosome level, we can then classify these ribosomes, each individual ribosome. Does it look more like this, or it looks more similar to another state, something like that? And this is called classification. And we can do this in 3D to get different states of the ribosome, let's say. So in this case, we could find, for example, fusidic acid inhibited ribosomes and some that are getting rescued and things like that. So from this, we would get multiple reconstructions. I want to quickly show how we prepare the samples so you understand the time limitation that we have. So commonly, the samples are prepared by what is called plans freezing, because we have to have a thin layer of ice, so we need to freeze the samples and it has to be thin. And for that, we commonly use a blotting robot. So we would have a freezing robot, sorry. We will have the sample that has to be applied to the sample carrier for the microscope that is called a grid. And this is not to scale. A grid is really small. I have one here. It's about 3 millimeters across. So it's really tiny. And we add a very little amount of sample, about 3 microliters. And then this is applied to some tweezers. And then we filter away most of the liquid because we need a very thin layer of ice. Otherwise, if it's too thick, the electrons won't penetrate and we will get black images, basically. And this has to be quickly frozen in liquid ethane. So to do this, the initial thing that I tried was that I had the fusidic acid locked ribosomes with EFG. Here, I added FUSP to rescue them, and as quick as I could add to the grid, then this would get blotted with the robot and then frozen. And from that, I could get as quick as more or less 25 seconds from adding FUSP, so 25 seconds of total rescue. And that proved out to be too slow, and most of the ribosomes were rescued, and I couldn't find FUSP, basically. So what we came up with is, what if instead of adding FUSP at this step, we go up to here. We set up the robot, we add the drop of fusidic acid locked ribosomes, we add FUSP here on the grid, and then do only these steps after adding FUSP. And from this, I could get as quick as 6 seconds. But in our preliminary results, what we actually found out is that, so this is a schematic to show it. After adding the drop with the fusidic acid locked ribosomes with EFG, we add the the drop of FUSP. But because it's so small, as I told you before, the diffusion is not fast enough, and the mixing, when you add with a pipette, doesn't mix completely. And we actually found that in different parts of the grid, there were different populations of the different states of rescue that we found. So actually this 6-second number that I say is the maximum contact time that FUSP has with these locked inhibited ribosomes. So it will be up to 6 seconds, basically. So from this, I process the data and classify the ribosomes, as I said, to identify the rescue states. And before going into the more interesting result, I first want to show you how the fusidic acid inhibited state looks. So I'm using the same colors as before, and this is one of the or two of the states that we can find in this data. And here we have the ribosome, tRNA, and EFG locked by fusidic acid. And if I show you where fusidic acid binds, this is the fusidic acid molecule, and it's bound between 3 domains of EFG here, close to what we call the sarsin-ricin loop of the RNA from the large subunit of the ribosome, that is here, and also a GDP molecule, because EFG uses GTP to function. And this, if we look at the 2 time points that we had, so what I said about mixing on the grid, and also the 25-second time point where I mixed on the Eppendorf before, we can clearly see the effects of the rescue, how this is a percentage of ribosomes that are in these specific states compared to a percentage of the total amount of ribosomes. You can see how it dramatically decreases over this time, these 25 seconds. And now let's look at the main result, which is capturing FUSP bound to EFG before they get released. You can see how FUSP here in pink is bound to EFG on green, and they are both sitting on the ribosome. And this, as I said, we couldn't find it at all. It was undetectable in this 25-second time point. But when we mixed on the grid, we could find a rare population, about 1.5%, that had FUSP bound to EFG. And if we look more in detail, you can see here FUSP bound to EFG. And this is where fusidic acid would bind. And you can appreciate that FUSP has absolutely no direct contact with fusidic acid when it's bound to EFG. And in fact, if we look at the cryo-EM density around this region, there is clear evidence that fusidic acid is still bound in this complex. So even though FUSP has bound to EFG, fusidic acid is still present and bound to EFG. So then how does FUSP release EFG from this inhibition? This becomes really clear if I show you now a video that I made in which we morph the structure of the locked EFG with fusidic acid to the one bound to FUSP. And you can see the changes that EFG encounters. You see this great conformational change of EFG, how it has to open up, it separates these two regions of EFG because FUSP is interlocking itself between them, and also how, if we go back, how this part of EFG is getting pulled out of the ribosome by FUSP, because FUSP is anchoring itself to the ribosome here. It's pulling away EFG. It's kind of prying EFG out of the ribosome. And when we compare the structures, we can see how EFG, now that is bound to FUSP, has much less contact area with the ribosome compared to when it was inhibited by fusidic acid alone. And this loss of contact with the ribosome will eventually make it free itself and release. So from this, we could explain the mechanism of resistance that was quite unclear before. So as I said, fusidic acid will bind to elongation factor G after translocation and lock it to the ribosome, not allowing it to leave, which it needs to, so new tRNAs can come in and deliver new amino acids. But in this case, in the FUSP type resistance, FUSP will be expressed. It will bind to EFG and pry it off the ribosome, basically. Make this huge conformational change that makes it lose contact with the ribosome and get released. And as I said, fusidic acid is still present, and it will likely dissociate once they are in solution. So this clearly explains how ribosomes can be rescued by FusB. But this was not all we found in this cryo-EM data. There were a lot of ribosomes that were in a different state, in which FusB was bound to them directly without EFG. Like you see here, FusB, the cryo-EM density of FusB in pink, bound directly on the A site of the ribosome. And these are really quite populated. In this early time point, they were almost 20% of the ribosomes had FusB bound to them. And after 25 seconds, it decreased slightly. And I will discuss this briefly later. And if we look more in detail, you can see how FusB is binding here close to mRNA at the decoding center, where the mRNA is decoded by the tRNAs, basically, and the ribosome. And very interestingly, we also found FusB bound at the A site, but mainly to the large subunit instead. So if we go back, here is at the bottom, bound mainly to the small subunit, and in this other structure is mainly bound to the large subunit. But bear in mind that this is much more rare compared to the other complex. And here, if we look more in detail, FusB spans from the tRNA that is on the P site up to the sarsin-ricin loop that, if you remember from before, I said that it was in close proximity to fusidic acid when EFG is present and inhibited by fusidic acid. And I started scratching my head because I thought it was a little bit weird that it releases EFG that is found here on the A site of the ribosome, but then it can bind independently of EFG where EFG was before, basically. So you can see it overlaps quite a lot to where FusB is binding to where EFG was. So it was a little bit puzzling that we saw this, and we are trying to understand what it means. So one of the things that we wanted to see is what is the affinity, how strongly is FusB binding to the ribosome? And to do this, we did fluorescence polarization. So I labeled FusB with a fluorophore, and this with fluorophores, when you excite, so you excite with some light, and they produce a different wavelength of light. And if you do this with polarized light, if the fluorescent molecule tumbles very rapidly in solution, it will depolarize the emitted light. But if it moves slower, then it will be slightly polarized. And then, because FusB is much smaller than the ribosome, we can track how it binds by tracking the increase in polarization. If it binds to a ribosome, which is like 10 times bigger, 100 times bigger, it will increase the polarization of the dye of the emitted light. And this is what I show here. This is in the x-axis, concentration of ribosomes, and we keep the concentration of labeled FusB constant. And then this is the polarization value of the emitted light. And you can see how it increases the polarization the more ribosomes we add, because FusB is binding to them. And from this, basically, the half point will be approximately the constant of dissociation, and it turned out to be 320 nanomolar. But knowing the constant of dissociation doesn't really tell us much if we don't know how much FusB is in the cell when it's inhibited by fusidic acid. So we wanted to try to answer this question. In this clinical strain that has FusB, how much it produces when fusidic acid is there. For this clinical strain, the expression of FusB, so how much is produced, is controlled by what is called translational attenuation. And this is by following the secondary structure of the messenger RNA that the ribosome has to bind to then translate. So what happens is in bacteria, ribosomes recognize what is called Shine-Dalgarno sequence, or SD, that I'm showing two here. And they have to recognize this, and then they can translate what comes next. So, for example, here is the start of the gene for FusB, and here is the Shine-Dalgarno sequence. And here is in a hairpin structure that is not available for the ribosome to bind. Whereas there is another Shine-Dalgarno upstream that is available. So normally the ribosome will bind to this one, and it will produce this leader peptide that is called. That is a small protein that is upstream of the FusB gene. But what happens is that in presence of fusidic acid, the ribosomes will not be able to produce this peptide easily. They will get stuck because EFG will get locked and they will get stuck in the middle. And when that happens, because the ribosome is stuck here, the secondary structure of the RNA changes, and these two hairpins that I show here merge together to form this hairpin. And then this Scheindler-Garno sequence for FusB will be open, and then other ribosomes can bind and produce FusB. So that's how it's regulated. So what I set out to do is to do relative quantification by mass spectrometry, because we wanted to know how much FusB is compared to EFG, because that would be enough information, and it's much less complicated than absolute quantification. So what I did is I took this clinical strain and I grew it to exponential phase in presence of 2 micrograms per milliliter of fusidic acid, which is quite above the minimum inhibitory concentration for wild-type Staph aureus, but this clinical strain that has FusB can grow. And then we sent it to the proteomic course facility in Gothenburg, and Karina Silblom did the mass spectrometry analysis, and she found out that the FusB to EFG ratio in the cell in these conditions was 1 to 2.3. So what that means is that for every roughly 3 molecules of EFG, there is 1 of FusB. And we know that from other published papers that there is about 10 micromolar of EFG in the Staph aureus, which means that FusB will be at low micromolar range, and ribosomes will also be at least the same concentration as EFG, which means that both ribosomes and FusB are well high above the constant of dissociation that I measured with fluorescence polarization, which means that in theory FusB could also bind the ribosome in vivo as well as binding to EFG. But I should say that the affinity of FusB to EFG is higher than to the ribosome, about 4 to 5-fold. So this kind of help us understand the importance of this binding to the ribosome. But now I want to show just the last thing about this CARIM dataset that I found that wasn't all. I also found another state of the ribosome where both FusB and EFG were bound together. But here, both FusB and EFG are in a non-canonical region out of the A site of the ribosome. And in fact, we can only see 2 domains of EFG, so about half of EFG, which means that the rest of the ESG is present. But since in each ribosome, if it's not interacting stably with the ribosome, it will be tumbling around in solution. So in each ribosome that we average to get the 3 reconstruction, it will be in a different position and it averages out as noise, basically. And that's why we can only see half of it. But since this is quite not very populated and is about the same population at both time points, and it's bound in a non-canonical region and fusidic acid is not bound to EFG, we concluded that this must be a rebinding state. So that because we have FusB, EFG bound together in solution, they may rebind in this way, but we don't think it has relevance in terms of fusidic acid resistance. So if we look back to the time resolution of this experiment, we have 2 time points, this mixing on the grid that is up to 6 seconds and then the 25 seconds from adding FusB. We can observe that population of all these different states. And as I showed before, the fusidic acid inhibited states greatly decrease after 25 seconds. And this is because FusB is rescuing EFG, and then it doesn't get locked again now that it's bound to FusB. The rescue state here that I showed before release was a small population and only detected in this early time point. And the FusB only bound states are quite populated at both time points, but they decrease slightly. And I think the reason for this is that we also have EFG in solution, and it's much higher concentration than the ribosome that we have. So FusB also is binding to EFG, not only the ribosome. And the more time, the more it will bind to EFG as well. So to conclude, just to remind you, fusidic acid inhibits protein synthesis or translation by locking elongation factor G to the ribosome. FUSB type resistance, which is called is a mechanism of target protection, is the most common in Staph aureus. And the way it works is that FUSB causes a major conformational change of EFG that breaks its contact with the ribosome. It kind of pries it off the ribosome, and it releases it that way. And I should really focus that FUSB does not interact at all with fusidic acid, and it doesn't need to cause its dissociation. Instead, it will just release EFG, even in the presence of fusidic acid, like we saw. And finally, that we found that in conditions of fusidic acid inhibition, FUSB is present about half to a third of EFG, and this means that it's quite high concentration in the cell. And finally, that we also found that FUSB can bind directly to a ribosome in absence of ESG, but the function of this is still unclear. So in the future, we should try to figure out what is the function of this? Why is it FUSB binding to a ribosome? And especially, why is it binding to the A site, which it just took out EFG from there, right? And maybe there are some clues in FUSB homologs, so other proteins that are related by evolution to FUSB and are similar, because some homologs are present in organisms that are unlikely to have encountered fusidic acid, and for example, Listeria or Enterococcus. And actually, other target protection mechanisms like tetracycline resistance proteins and ABCF type resistant proteins have evolved from housekeeping proteins that were doing a normal function in the cell, and then they happen to evolve to give resistance. And for example, in the case of these tetracycline resistance proteins, they evolved actually from elongation factor G, which is really interesting. So it could be that it's the same for FUSB. It was an accessory protein to the ribosome, and it somehow provided resistance to fusidic acid. But of course, this is all speculation. Another thing that I think we should point out is that how could we counter FUSB type resistance? Because since it doesn't interact with the antibiotic and it doesn't need to release it to give resistance, then, for example, derivatives of fusidic acid are unlikely to work since, as I said, it doesn't interact directly and it doesn't need to release it. But maybe some strategies that target other parts of EFG or that prevent that FUSB binds to EFG could work, or somehow increasing the contact of the antibiotic with the ribosome as well. And finally, I just want to acknowledge Maria's group and also our collaboration with Suparna Sanyal that also participated in this project, and Karina Silblom from the Proteomics Core Facility in Gothenburg. And of course, thank the UAC for funding and all the other funding agencies that made this project possible. And this is in the later stages of review, but the preprint is available if you're interested in more details. But I think it will be published soon, hopefully. And thank you all for listening. So I'm open for questions.
Thank you so much, Adrián. What a wonderful investigative project. The deciphering, how this mechanism works, which I, until you started working on your project, I didn't know that. We didn't know so much about how this actually worked. We just, knew that it was doing something, and now we actually can look tiny enough to actually see it. So, Philip, you have your hand up. Do you want to ask Adrián something directly?
Yeah. Great job, and it's a really exciting story. I have like two questions on slide 12, when you were showing on how you're doing classifications. To my understanding, you are now looking at the structure, sort of de novo, something that kind of hasn't been done before. So how do you know for a structure that hasn't really been resolved before, or something that you haven't seen? How do you know in which class it's going to belong?
Yeah, usually what I do is I get a consensus with all the ribosomes I have, right? And then this can be classified. For example, you want to classify over one region that you are of interest. In this case, I classified over looking if the particles contain EFG, for example, something like that. So I looked particularly at this region to see if FUSB was present, for example. And this will be done by some algorithms that will identify for this particle, is it different than this other one? And when many of them are similar, they are combined together to make a 3D reconstruction. And that will be the average of those that are classified together. Because obviously from only one particle, one 2D image, you cannot get a 3D reconstruction. You need thousands of them. But the thing is, it will compare each of them basically. And if they are similar, they will be grouped together, and this will be done iteratively. And once you have you finish the classification, you can get many reconstructions in a simplified manner. I think this is how I can explain it.
And the second question is, you showed very nicely how FUSB unlocks EFG from the ribosome. Is that energy dependent mechanism so that FUSB changes like some sort of conformation, or FUSB structure is pretty rigid? That just comes, takes things out, takes EFG out, and then it continues to do its job.
So FUSB doesn't utilize any energy molecule like ATP or GTP or nothing like that. And for example, EFG has used its GTP already, basically at this stage. And it looks like it's doing this by, I can go back to the video if you want. It's easier to see there. What we think is this domain, because of FUSB, these helices are getting like shoved between the domains of EFG. It makes it open up. That's one of the things. And here with the C-terminal domain of FUSB, this part is anchored to the ribosome in this position, but it's also interacting to EFG. And we think that's how it's pulling it out of the A site here.
Okay, very
Does that help answer the question?
But so FUSB is not like protein that is moving quite a lot, at least.
No. I don't think so. But of course, here we only see these two states, right? That I'm comparing. This movie, I'm making an interpolation, basically, with a program. We don't know exactly how this happens. This is what we can derive from seeing these two structures.
Yeah, very cool. Thanks.
Yeah.
Very, very nice, Adrián. One question came up to me when we were looking. There's a lot of steps on the different dynamics of this, so forgive me if I didn't get completely everything. But does FUSB when it comes out and exits EFG, together, then it does actually disassociate again, so that FUSB is available to do another round?
We think that's unlikely because the affinity between FUSB and EFG is really high, like 60 nanomolar.
So it kind of gets like
constant
trapped
dissociation.
completely there.
So once they get out together, they probably stay together. Yeah. But we don't know, for example, if this complex can do its job. Can it translocate? We don't know. It's something that is not so easy to test because it's difficult to know if you have leftover EFG that is free and is doing the translocation. What I have not shown here is the experiments that Suparna's group did, but we saw that in the presence of FUSB, the translocation step that the EFG is performing is slower. So probably either when they are bound, they are inactive or they have lower activity. But we cannot tell from those experiments.
All right. So, okay. So it means that maybe the translation step will be slower. So translation will be slower, but there's still...
I mean, the thing is, you still have a lot of EFG in the cell, right? As we saw, FUSB is quite high concentration when it's under inhibition, but also because it's regulated by this translation attenuation, I guess you get a spike when you have the inhibition, a spike of FUSB, and then it will be decreasing over time. And you still have more EFG than FUSB, so the leftover EFG can perform the function. The main thing is you want to get these ribosomes unstuck, basically, because it's not good to have one ribosome stuck. It's not only that, but other ribosomes can collide, for example, with that ribosome and things like that.
it gets a bit of a party in there. Yes,
Yeah.
Philip?
One question that you brought back with the evolutionary part. Why
Yeah.
do you think is that only gram positives have FUSB and not gram negatives? I mean, the the consensus in the community is that E. coli and gram negatives are inherently resistant to fusidic acid, which
Yes.
still is some sort of a debate. But why do you think that this wouldn't appear in gram negatives and it's only
So
in
your
gram positives?
question is kind of like two. First, I want to say that
Mhm.
gram negatives are inherently resistant. Not all of them, but most of them are inherently resistant to fusidic acid. But this is due to one, permeability, because the gram negatives have an extra membrane on the outside. And also, for example, in the case of E. coli, they usually have pumps. And E. coli has this Tolsi ACRB that has been shown to pump fusidic acid, or at least to bind to it. So that's why they are resistant. But to answer the other question, sorry, we don't know, but I have not found any FUSB homologs in gram positives, in gram negatives. But in gram positives, you can find in a lot of them. So, I mean, it could be what it has been said before is that it could have been some kind of gram-positive specific ribosomal accessory protein. I mean, there are other examples of other proteins that are act on translation are exclusive of either gram-positives or gram-negatives. But I cannot tell anything else, basically.
Cool, very cool.
Yeah.
Remind me one thing, Adrian. This I should really know, but I don't remember right now. Are gram positives and gram negatives the classifications that are evolutionarily relevant? Which means that are
Yeah,
they paraphyletic or because
I
I
think,
don't
yeah,
remember.
it's not so easy, I think, to look at it in that way, actually.
Yeah, so you cannot just say a clear cut, that this was
Yeah,
a
exactly.
housekeeping gene that maybe got another
It's
function earlier in the trio evolution or.
Yeah, exactly. But I mean, when I look at proteobacteria, for example, I cannot find any FUSB homologs.
Yeah, so something else is going on there. Yeah, really cool. Well, anybody has any more questions? If not, then we are going to first thank Adrián for presenting your research and your results. Really cool. We'll read the
Yes, thank
paper
you.
as soon as it's out as well. And Philip is being celebratory right now. And I hope that you guys enjoyed today's webinar. Valeria, Adrián, thank you for sharing your work with us.
Thank you.
And I hope to keeping track of what's going on with your projects. Thank you, everybody, and have a lovely
Thank
weekend
you.
ahead. Bye-bye.
Thank you. Bye.